Hello and welcome to Reviewing with Mrs. Wages. Today I'm going to talk about cellular respiration, glycolysis, and cell transport. Let's begin with objective number 1, which asks you to explain different cellular respiration pathways. First of all, the purpose of cellular respiration is to convert glucose into ATP. And if you remember, ATP is produced mostly in the mitochondria. This molecule stores energy used by the cell for life processes. And who does cellular respiration? Well, all living organisms do cellular respiration because all living organisms need ATP to stay alive. That means that both plants and animals do cellular respiration, as well as other living organisms. Now, why do organisms need all this ATP? Well, ATP is required for making essential macromolecules like carbohydrates, lipids, nucleic acids like DNA and RNA, and proteins. This picture shows you the equation for cellular respiration. The reactants are glucose and 6 molecules of oxygen. They get turned into the products, which are 6 molecules of carbon dioxide, and 6 molecules of H2O, which is water, and a bunch of ATP. Now, if you look at this overview of cellular respiration, it looks very, very busy. But in reality, there's really only 2 pathways. The first pathway is aerobic respiration. It begins with glycolysis and finishes in the mitochondria. The second pathway is anaerobic respiration, which also begins with glycolysis and finishes in the cytoplasm. So no matter which pathway you're going to take, aerobic or anaerobic, it all begins with glycolysis. This green box shows you an overview of glycolysis. Glycolysis begins with a molecule of glucose, which gets converted into ATP, pyruvate, and NADH, and this all happens in the cytoplasm. Next question is critical. Is there enough oxygen available for aerobic respiration? If the answer is yes, then pyruvate and NADH will head to the mitochondria to do aerobic respiration. Aerobic respiration finishes in the mitochondria with these 3 processes: converting pyruvate, the Krebs cycle, and the electron transport chain. The goal is to make a lot of ATP. That's the benefit of going by this pathway, is because when oxygen is present, a lot of ATP can be made. However, sometimes oxygen is not readily available. In this case, pyruvate and NADH will stay in the cytoplasm and finish out fermentation, which is a form of anaerobic respiration. And there is 2 types, alcohol fermentation and lactic acid fermentation, depending on the kind of organism you are. The benefit of this pathway is not to produce ATP, because it doesn't. What it does do is it provides NAD positive so that glycolysis can continue making a little bit of ATP until more oxygen is available. Now let's consider 2 types of fermentation. I'll begin with alcohol fermentation. Yeast and other microorganisms carry out this process. It occurs in the cytoplasm following glycolysis. The reactants for alcohol fermentation are pyruvate and NADH, which, as you know, were produced in glycolysis. The products are NAD positive, which is needed to keep glycolysis running, carbon dioxide, and ethyl alcohol. Another type of fermentation is called lactic acid fermentation. This occurs in the muscle cells of mammals. It takes place in the cytoplasm of the cell. The reactants from glycolysis are pyruvate and NADH. The products are NAD positive, again, to make glycolysis to continue making ATP, and also lactic acid. No carbon dioxide is produced by this process. Now let's consider objective number 2, which asks you to explain glycolysis. Glycolysis, as you remember, begins both aerobic respiration and anaerobic respiration. This process starts with a molecule of glucose and is located in the cytoplasm of cells. The very first change we'll do to glucose is to bring in a couple of ATP. Each one will drop off a phosphate on each end of the glucose molecule and leave as ADP. In this way, we're burning a couple of ATPs at the very, very beginning. Step 2, that very cumbersome molecule, phosphates on it, now breaks apart into 2 separate smaller molecules. In step 3, we're going to bring in some NAD positive, which will steal 2 electrons and 1 hydrogen from each of these half molecules and form 2 NADH molecules, which will go on to either the electron transport chain in the mitochondria or continue on to fermentation. In step 4, enzymes will attach inorganic phosphates to each of the half molecules. At this point in step 5, 4 different ADPs can come in and steal those 4 phosphates. When they do, they'll create 4 ATPs. Now remember, even though we're making 4 ATPs in this step, we literally had to burn 2 at the very beginning. So there's only a net gain of 2 ATPs from glycolysis. And finally, some more enzymatic changes will convert the last 2 molecules into our final product, which is 2 molecules of pyruvate. In summary, the reactants of glycolysis are glucose, ATP, NAD+, phosphates, and ADP. The products of glycolysis are ADP, NADH, ATP, and pyruvate. Now let's consider objective number 3, which is a flashback to cell transport. How will cell transport be carried out in this example? Well, that depends on the concentration of the solute. In this case, the solute is sugar. Inside of the cell, there's 9% sugar. Outside of the cell, the concentration is only 7%. That makes the inside hypertonic, and the outside is hypotonic. Now remember, the words hypertonic and hypotonic are always referring to the concentration of the solute. They're not referring to the concentration of the water. If the membrane is permeable to the solute, sugar, then the solute will move from inside of the cell to the outside, from high concentration inside to lower concentration outside. This is the same thing as saying that the solute will move towards the hypotonic side in this direction. If the membrane is also permeable to water, then the solvent, which is water, will move to the inside of the cell, which is the hypertonic side, in this direction. Remember, the goal of both diffusion and osmosis is to achieve equilibrium. If the concentration inside matches the concentration outside of the cell, the areas are said to be isotonic. And once the concentration on both sides of the membrane are in equilibrium, both the sugar and the water will continue to move back and forth across the membrane, maintaining equilibrium. That's all for now. I hope this helps, and as always, happy studying!